Differential expression and signaling activation of insulin receptor isoforms A and B, a link between breast cancer and diabetes. This research was published on the National Institutes of Health website by Loma Linda University. It grabbed our attention as the paper was conducted on a link to cancer, a popular medical topic among scientists and general public. They suggest a link between breast cancer and diabetes by differential expression and signaling activation of insulin receptor isoforms A and B.
The possible link was identified whilst trying to explain why it is that the death rates of breast cancer remain higher in African American women when compared to Caucasian women. The following factors are being explored as causative agents: differences in access to healthcare, different utilization of screening tools, and biological factors. As such, these people address this issue by studying the insulin receptor expression and the insulin-like growth factor activation of this receptor signaling pathway in breast cancer cells and impaired breast tissues from both ethnic groups. The insulin-like growth factor, also known as IGF-2, is one of three protein hormones that share structural similarity to insulin. It plays a key role in the regulation of growth and metabolism, as well as in the initiation and maintenance of breast tumors, meaning that a high IGF-2 level increases breast cancer susceptibility and stimulates tumor growth and progression by signaling through the IGF-1 and insulin receptors. Alternative splicing of the insulin receptor gene results in two transcripts, insulin receptor A and insulin receptor B. As mentioned, the tumor tissues and cell lines from African American breast cancer patients express significantly higher levels of IGF-2 and higher IGF-1 receptor activation as compared to Caucasian women. Since IGF-2 also activates the insulin receptor, they suggest that the higher IGF-2 levels among African American breast cancer patients translates into higher insulin receptor activation. So a bit about the insulin receptor. It is a heterotetrameric protein consisting of two extracellular alpha subunits, known as ligand binding, and two transmembrane beta subunits, known as tyrosine kinase domain, that can be activated by insulin or IGF-2. Insulin receptor A is activated by IGF-2 and insulin to promote mitogenic and anti-apoptotic signals. However, IGF-1 does not bind the insulin receptor A. Insulin receptor B activation induces cell metabolism and differentiation signals. The activated insulin receptor B subunits phosphorylate several intracellular substrates, including the focal adhesion kinase, which is involved in the integrin and growth factor receptor signaling pathway. Therefore, the present study focuses on the differential expression of the insulin receptor isoforms A and B and how IGF-2 expression relates to phosphorylation and activation of the insulin receptors as part of the mechanisms associated with more aggressive breast tumors and increased mortality observed in African American women.
Breast tissue specimens were taken from paired frozen breast cancer and normal tissue specimens from women with ages ranging from 20 to 90 years. The samples containing cancer were stage 2 or 3 and affected the ducts or the papillary thyroid gland. Cell lines established from tumors were maintained in a 5% CO2 incubator at 37 degrees Celsius. They were then supplemented with penicillin, streptomycin, glutamine, amphotericin, and fetal bovine serum. Cell lysates were collected, centrifuged at 800 rpm for 5 minutes and kept frozen at -20 degrees Celsius until assayed. Membranes were then incubated overnight at 4 degrees Celsius with an anti-INSR alpha subunit, a rabbit polyclonal antibody that recognizes INSRA and INSRB, purified rabbit polyclonal, and an anti-human INSRB antibody. After 3 10-minute washes in the PBS tween, the secondary antibodies were added to the membranes, followed by 3 more 10-minute washes and incubation with HRP complexes, after which the proteins were visualized. The RNA was extracted using a tri-reagent. The frozen tissue sample was homogenized in the tri-reagent, and this RNA was kept at -80 degrees Celsius until assayed. Reactions were performed in a mixture containing 1X SYBR green supermix, PCR buffer, primers, multiscribe reverse, transcriptase, and an RNA inhibitor. Fluorescence was attached at the end of every 72 degrees Celsius phase, and to exclude the contamination of nonspecific PCR products such as primer dimmers, a melting curve analysis was applied. Statistical differences between mean values were determined by using one-way analysis of variance, paired t-tests and independent t-tests. The values were then expressed as a mean, and a value of p higher than 0.05 was considered significant. The phosphorylation of insulin receptors in African American cells was higher compared with Caucasian cells. The insulin receptor Western blot was also used to proceed with further analysis of the insulin phosphorylation of tyrosine 1162-1163 to help stimulate receptor intrinsic kinase activity. In contrast, the estrogen receptor in African American cell lines express higher levels of insulin protein compared to Caucasian cells.
The Western blot shows an analysis of total insulin receptor and insulin receptor B expression in breast tissue samples. To the extent antibodies were also used in these samples. The insulin receptor antibody D59 recognized as an alpha subunit of insulin receptor A and B. The premenopausal AAM expressed significant level of insulin receptor protein when compared to the premenopausal CAM samples. Furthermore, the premenopausal CAN and CAM samples had shown a high increase of insulin receptor B isoform as compared to the AAN samples. The postmenopausal of African American and Caucasian women insulin receptor expression had shown an increase in AAM when comparing to CAM tissue samples. This also indicates that the result receptor B protein levels were higher in CAM samples compared to AAM breast tissue samples. Breast cancer can be influenced by numerous factors such as environmental factors, culture, genetic, lifestyle, and socioeconomic. Breast cancer in African American continues to show high mortality rates with younger age distribution compared to Caucasian American women, and there is still limited understanding in translating the effective intervention to reduce mortality rates. The research demonstrates that the insulin growth factor 2 plays a major role in contributing to a higher level of breast cancer mortality in African American women.
Insulin A upregulation is associated with increased insulin growth factor 2 signaling, whereas insulin receptor B upregulation is associated with insulin-mediated metabolic effects. Thus, insulin receptor A expressing cells undergo proliferation when stimulated by insulin growth factor 2, whereas they preferentially activate glucose uptake when stimulated with insulin. The results show that insulin receptor A, insulin receptor substrate, and FAK changes observed in breast cancer cells correlated with results obtained from breast cancer tissues. Thus, this data suggests that changes in insulin growth factor insulin signaling pathways are maintained in cells established from primary tumors. Levels of hybrid insulin receptor and insulin growth factor have been shown to be increased in breast tumors, leading to increased affinity for insulin growth factor 2 over insulin. Therefore, the co-expression of the insulin receptor A and insulin receptor B isoforms increases the hybrid receptor formation, reduces the metabolic effect of insulin receptor B, and promotes mitogenic and survival signals by insulin growth factor 2. It is in fact insulin, not insulin growth factors, that stimulate tyrosine phosphorylation with consequent FAK dephosphorylation and mediation of insulin metabolic effects through the insulin receptor B, yet in contrast it was also observed that FAK signaling cascade was actually highly activated in the ERPR African American cell line by insulin.
Epidemiology studies shows type 2 diabetes and metabolic syndrome is associated with increasing breast cancer. In a transgenic mouse model, overexpression of IGF-2 leads to insulin resistance and eventually to type 2 diabetes. Thus, increased IGF-2 in normal African American compared to Caucasian may increase cancer but also cause higher breast density, more aggressive and rapid breast cancer progression, insulin resistance, and type 2 diabetes. The different activation of insulin receptors between African American and Caucasian coupled with IGF downstream signaling pathways may contribute to the increase of aggressive breast cancer phenotypes observed among African American breast cancer patients. IGF-2 insulin receptor A and B isoforms play a major role in the onset and progression of breast cancer, meaning better understanding of the receptors in different populations can lead to new ways to cure.